All right, now we're going to look at cooperativity. All right, so cooperativity involves, before we actually get started, is the actual carrying capacity of the hemoglobin molecules that we learned before, right? So what was hemoglobin? So hemoglobin was this globular protein, right? So you have this globular protein that is surrounded by this heme group. In the heme group, we have this iron, this ferrous deposit with a positive 2 charge, right? Main function, bind to oxygen, right? So the main function, as we saw before, what specific cell types carry this hemoglobin? Red blood cells, so red blood cells or erythrocytes. So you have your red blood cells coming in here, and they possess the specific hemoglobin molecule as their main function to carry oxygen to the tissue of origin, right? And so we have oxygen molecules coming in here. So if you remember oxygen has a partial negative charge, right? And the iron here, you can see that it has the partial positive charge. So these molecules are attracted to each other. And how many molecules can they bind to? How many oxygen molecules can the iron bind to? 4. So there's 4 subunits, right? But in the red blood cell, there's an abundance of these hemoglobin, right? So it's not just one hemoglobin for red blood cell. That would be the most abnormal problem, right? There are problems that have this situation, right? We call those anemias, right? So iron deficient anemia would be a perfect example of having lack of these iron groups for the oxygen binding. All right, so this main function is here, right? And so as these oxygen bind, right, we can graph it on this little axis here. So there's this, the cooperativity is basically a measurement of how the oxygen saturation takes place with the hemoglobin, right? So if we look at on the x-axis, we have millimeters of mercury, right? So the partial pressure of oxygen, right? And here on the other type is percent saturation. So what does saturation mean? So saturation means the amount of actual hemoglobins bound to this molecule, right? So if I were to make a little equation here that represents the oxygenation or reduction of hemoglobin, is set off here. So hemoglobin comes in its reduced form. So reduced form meaning it has the hydrogen, goes in the alveoli, there's an abundance of oxygen. This abundance of oxygen causes for this oxidation to take place. So now hemoglobin here becomes oxyhemoglobin, and you have this hydrogen group that is present here, okay? This equation is reversible, meaning go back and forth depending on the site of location, right? Going from right, actually let's not say it like that because then it'll be confusing with the right or left. Going from a reduced hemoglobin to an oxidized hemoglobin, this direction here in blue, this happens in the alveoli. When the hemoglobin becomes deposited into the actual tissue and it releases the oxygen, this direction, this happens in the systemic tissues. Right? That's why it's reversible, because both of these are constantly happening throughout the systemic and pulmonary circuit, right? So going towards the oxygenated hemoglobin, happens in the pulmonary circuit, and then going into the reduced circuit, reduced hemoglobin, that happens in the tissue or the systemic circuit, right? And so cooperativity, the way it's measured is through this sigmoidal curve, right? So you see this sigmoidal curve and you see this saturation. This saturation is measuring this oxyhemoglobins, right? So the number of these oxygenated hemoglobins, right? And so the reason why it's sigmoidal is hemoglobin's actual structure, when it's not bound to any oxygen, right? So it's in this reduced form, is in this T state, or quote unquote tight state. So that what that means is that its actual receptors and iron groups are not easily as available for the oxygen to be bound. However, when one oxygen binds, this reconfigures and it becomes into this state called the R state. So it goes from the T state to the R state. So this is the more relaxed state. right? And again, I'm gonna write this both arrows, meaning it's reversible, right? So if you lose oxygens, you lose cooperativity. When you gain oxygens, you gain cooperativity, and so now when you're in this relaxed state after the one binding of the oxygen, now all these molecules are available, nicely available, and specifically the molecule I'm talking about, the iron groups, and now the oxygen can now easily come in and bind more. So more oxygen with the partial negative charge can come in much more freely to combine to these iron groups. Now, when you have all of the 4 sides bound, you have pure saturation, right? Meaning that there is no more availability of the oxygen to bind any other receptors because there's no more stuff, right? And so that's where you have this actual plateau right here. This is saturation, where it doesn't matter when we look at the bottom x-axis here of the graph where it's showing you the concentration or the partial pressure of oxygens, right? So as you go up higher, 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 whether it's from 0 to 25 to around, this is about 75, and then eventually to 100, it doesn't matter if you keep going to 120, 140, 160, there will be no more hemoglobins binding, right? Because everything has already been spent, right? And so cooperativity, again, is when there is one binding of this oxygen to this ferrous group, it changes from a tight state to a more relaxed state, okay? And so the receptors become more available, readily available, in order for the oxygen to bind. And that's why you see this exponential growth of saturation, right? So this exponential growth is what is the whole phenomenon of cooperativity. So if I draw a little diagram here, this is the whole cooperativity. And this is the general mechanism of how these hemoglobin molecules are able to create such a very realistic pinpoint of transferring oxygen to the site of interest.